So in the last class, we were discussing about several techniques, including of which we had a focus on RT-PCR. So RT-PCR, we had two ways. It is easy to get confused with that. Is it referring to reverse transcriptase PCR or is it referring to real-time PCR? The real-time PCR is much more sophisticated and also valuable. And reverse transcriptase PCR can only tell you if there is an expression of a particular gene or not. Right? It cannot say how much of this expression is present or not. Whereas real-time PCR, which you can also say it as qPCR, Q for quantitative PCR, okay? So it can tell us about the amount of product formed in each type of cycle. And for that, if we can say after each round of PCR cycle, how much of product is being formed, if we are able to quantify, that means we can quantify how much of the sample is, the template is present. The principle is simple. If we have more template, then we will be able to detect it early. In the PCR, in the RT-PCR machine, we will be able to detect it early detection. Low template, amount of template is low template, then delayed detection. Right, the signal for it, as we will see in the next slide, there is delayed detection. So there are two detection mechanisms that are used. One is cyber green. It's a fluorescent dye that binds to double-stranded DNA. So the amount of double-stranded after each round, say 2 templates will become 4, that might become 18, 8, 16, 32, and so on. It will double each cycle. So you'll also, that means the amount of double-stranded DNA is doubling each cycle. So cyber green will increasingly bind to double-stranded DNA and fluoresce. Okay, that can be detected. It is an easy one. It is less sensitive than compared to the other one, Tachman probes. Tachman probes have two things. One is a quencher. That's a molecule that reduces the fluorescence of the reporter. Reporter is a fluorescent molecule, but if it is within close proximity of the quencher, it cannot fluoresce. But if somehow this probe gets degraded and the reporter is somewhere else here, for example, then it will fluoresce. Okay, so we have to use we have to use two primers, forward primer and a reverse primer, and then we should also use a probe. The probe is a nucleotide that has homology or complementarity to the target sequence like this. On one side it has a reporter, and on the other side it has a quencher like this. So DNA replication, the DNA polymerase should have 5' to 3' exonuclease activity. So the DNA polymerase will synthesize this way, and it might come into the Tachman probe is obstructing its way. So it will chop this DNA like this, and will keep on moving forward. Right, and because of the cleavage of the probe, the reporter now is free and away from the quencher, because of which fluorescence will happen. So after each round of PCR, we will have more and more of these probes cleaved. That is indicative of the double-strand DNA formation or the target amplification. Okay, which is indicative. So it is like signal is indicative of the polymerization or the target sequence formation, which is indicative of the template in that particular cycle. which is again eventually indicative. The amount is indicative of the template in the sample, right? So it works on that principle. So if it generates the computer, the machine will read all the PCR. We call it real time because as the number of cycles is happening, it keeps on reading after each round of cycle how many products are formed. That is based on the reporter intensity, the signal. And every machine has a detection limit. So based on that, uh, if we have more number of templates, that template is going to develop faster and is going to come to be detected early, right? And then it will have a kind of exponential thing, and then it will reach a plateau. The same thing if something has lesser amount, then it will show it delayed CT value. And if you have the least, the sample containing the least amount of template will show up a curve at the last. Okay. So based on that, we can relatively say that A has the highest amount of particular mRNA or anything in the DNA. It can also be measured. So A has the highest, followed by B, and then C has the least. So real-time PCR can quantify and tell us if a gene is present or not, if a RNA is present or not, and it can also tell us how much of it is present relative to some other things. I hope you know that for COVID analysis, they always do. There are two different types of something like quick test, rapid test. Yeah, that is the correct word. Rapid test, and the other one is RT-PCR based test. RT-PCR based tests are much more reliable. There is about I think somewhere around 40% reliability is 40% less, or false negatives are being given. That means even though the patient is positive, they are giving false negative, and that is going to cost not only that person's life, but that person may also infect multiple other people, assuming that he is negative, right? This happens many a times because of these kinds of rapid tests. But in the current situation, that is the best we can do, because it also highlights why the disadvantages of RT-PCR. You need time. Real time, the probes are actually can be expensive, and cybergreen is much cheaper. But you also need a sophisticated instrument, right? These are some of the problems in a pandemic like this. It's very difficult um, to analyze a large number of that much number of samples. But otherwise, RT-PCR can recognize, can test about 96 samples in one run, which can be between say 2 to 3 hours. Okay, and then the reading has to come. So somebody has to collect sample. The number of samples received is too high, and that is one of the problems with RT-PCR. But rapid test is much more easy. Readily, anybody can do that. You don't need technical skill to run the RT-PCRs. Yes. The next technique is about RNA in situ hybridization. In situ is within the cell. You can think of it like that. So why do we want to do this is you want to know if particular RNA, where is it located, if it is there or not. That is one thing that can be told. And the second thing is if it is where is it located within the cell. Okay, especially in a tissue, if we have, and in the tissue we want to know which cells are expressing a particular RNA and which side of the body and so on. If one wants to do that, RNA in situ hybridization is a good one. So basically, the idea is to detect the mRNA. So we have a complementary DNA, so that it's a single-stranded DNA. It already has some labels attached to it. Okay, deoxygenin label is attached, so that at a later time you can have antibodies to deoxygenin. Um, Deoxygenin. It's difficult to pronounce, but yeah. So we should have permeabilize the membrane. Otherwise, the RNA cannot enter. The DNA cannot enter into the cell. So using detergent, um, one can allow the uh, pores to form, because of which the membrane, the complementary DNA is enters into the cell and base pairs with the mRNA based on the concentration, if it is there or not, and how concentrated it is. Sorry. And then we add antibody, which is tagged with alkaline phosphatase. This antibody is against deoxygen, and then it binds to that, and now we have an alkaline phosphatase to which we can add a substrate that is colorless, but sorry, but because of the action of alkaline phosphatase, that is the label on the antibody, it will convert the substrate into a colored compound. Okay, just give me 1 second, I'll clear my throat. Sorry for the interruption. So you can also see the same thing in the next slide, which is almost the same. I don't have to take it in that way. But I just wanted, I'm using this so that you can think of this particular part. Antibody can be anti-G-toxin. The tag that we have deoxygenin, or you can also use avidin with a fluorophore. Or you don't have to have all the time alkaline phosphatase and then add a substrate and then you get a colored compound. You can also have it as a link to a fluorophore, so that directly it will fluoresce and you can observe it under the microscope, and then we know where, in which cell, in which tissue, and in which cell this particular gene or RNA is being expressed. Okay. So the next technique is called as primer extension. Say, let me associate this RNA in situ hybridization. The principle of this is by hybridization between the probe and the RNA. And this is, we are trying to hybridize the target RNA in the cell. Okay. And we are trying to, hybridization of RNA in the cell. That's what you can think of. And once you know that, and if you know the detection systems, it's over, right? Yeah. So here is primer extension. I think somewhere we had a technique called race. I don't know if we have finished or not. There are two different race, 5' race and 3' race. I think I will finish it some other time. If I didn't, please remind me. So if I'm trying to work on a gene, and I know that this is your starting reading frame and so on, I want to know which is what is +1. I hope in some of the introductory classes I described about the structure, general structure, structural features of a gene, right? We have a promoter, and the promoter, the +1 is the site where transcription starts, transcription of the mRNA. Promoter is the bind where is the site where all the transcriptional machinery would have bound, and then the first base would have been added. That would have been referred to as +1. From here, if you count, you will get the -10, right? That is TAT, AAT in E. coli. I hope you remember -10 and -35 sequences, TTG, ACA. So somewhere base there will be a ribosome binding site signal for it. In eukaryotes it is called as Kozak sequence. In prokaryotes it would be called as Shine-Dalgarno sequence. This is where the ribosome would bind. And then we have the start codon, that is ATG, that is AUG here, and that is where translation would have started, right? And the first amino acid would have been methionine would have been added, and slowly a protein would have been generated. And then there would have been a stop codon, say TGA, and that is where it ends. And there would be a terminator, typically, not always. If there is a terminator, that is the end of the transcription. So keep this in mind. And in a situation where I want to find this is the 5' side, right, of the mRNA. This is the 3' side. I want to find the +1. Where does transcription of this particular gene start? That's my question. And how can we find the answer for it? It is actually a simple method. It's almost similar to that of what we would have done for. It has some high similarity with the, or it is in conjunction with the Sanger's method of sequencing. So what you take is, if there is an mRNA like this, and my target is to find out the first base in the mRNA, and because I know the gene, I will take I'll make a primer that is complementary to this, and then I will do reverse transcription. Okay, and reverse transcriptase will add until this, and it will stop. And if you are running it based on, if you are running a gel, vertical gel, polyacrylamide gel, then you would have it run at a certain length. So, but how can I say what is the sequence or base of it? Where exactly does it start? Say if I use the same primer and perform Sanger's sequencing, right? So it will give me the whole length. Say, for example, I think primer would have started somewhere here. I'm using the same primer for this part also, right? So in the Sanger's method, it would have generated so many fragments of different sizes like this. And in the primer extension method, assume the primer has generated something like this. What would that mean? This is same, and this would mean that this is the sequence identical to it, right? All these, in Sanger's method, all these reactions, when you run the electrophoresis, they are getting resolved to one base difference. The difference between this fragment and this fragment is just one additional A, and here it is one additional T. That is how we were able to read the sequence in the first place. So if you have used the same primer, and for same primer extension as well as for sequencing, then whichever is parallel to this or running at the same length indicates that is this particular sequence from down here, right, as the same sequence of the mRNA as well, because we are using the same primer and the length is same, which would mean that way. Typically, a reaction would primer extension reaction would look like this. So we were doing some experiments. So I did. This is the sequencing PCR, and here we have, this is the end of the mRNA. Okay, this is the end of the mRNA. At this in this experiment, you are trying to see if the mRNA is broken or not, and because in some conditions we don't need to know the experiment. I hope you see that there are bands which are indicative that DNA is being sorry, the mRNA is being cleaved at that location. Okay, you don't have to remember that, but you should know that every time PCR is done, and if you want to know the sequence of this particular site, right, then you should have done the primer extension along with the Sanger sequencing with the same type of template. Here you're taking the DNA template, DNA of the whole gene from here to here, of which mRNA would have been a smaller one. And you're using the same template, and you're running the gel of the reaction, the sequencing reaction as well as primer extension in the same at the same time, so that you can compare and detect the sequence of the 5' side of the mRNA. So it is called as primer extension method, because you're just like in the, I hope you remember this method also. We use something like primer extension method in site directed mutagenesis, right? When we have a mutation there, we just add the mutation. When you want to make it here, we include that mutation in the primer, so that whenever whichever is all the DNA molecules that are formed, most of them will have the mutation. There we call that as primer extension method. Here it is primer extension assay. Okay. Subtractive hybridization. I think you remember this. The goal of it is to find what is expressed in A, specifically A, right? So A minus B should give the difference. We are subtracting, this would be the tester, and B would be the driver. So we are subtracting all those that are common in both the types of cells. Say for example, whichever is expressed when you remove, say for example, A is producing some mRNAs called as 1, 2, 3, 4, 5, and B is producing 2, 4, 6, 2, 4, 6. And if we subtract, we will get 1, 3, 5. Is that it? I hope so. So we are subtracting the genes expressed in B from the genes expressed in A. So we will get these, right? That is called a subtractive hybridization. And this we are able to subtract by hybridization. We will see that we have already seen, but we'll just go through it. The driver is the one that we are subtracting from tester. Tester is the A here. And we are deducting or removing whatever is produced in B also, common ones. So the driver will usually have biotin, and cDNA is prepared for both of them. Only thing difference is the driver mRNA will have the biotin added. Then we mix it up, denature, and reanneal. When we reanneal, we do it slowly. So there are 3 possible combinations. The driver and driver mRNA or cDNA can anneal like these. Some of them, which are common in both driver and tester, would form something hybrid like these. Those tester mRNA that do not have any complementary from the driver will reanneal like this. One, we should also think that remember that I think about 10 times more of driver is taken. Okay, so that because there is also possibility from normal those that are common, the tester mRNA can also, or cDNA can also base pair back like this. But when you include 10 times more of driver, the probability of these will be reduced. Only those unique tester cDNA which do not have complementary sequences in the driver cDNA will bind together RNA like this. So if you pass all these mixture of cDNA through a column consisting of streptavidin, streptavidin has affinity for biotin. All those that are having biotin will get stuck or retained in the column, and the rest of the cDNA will pass through the column, right? So those with a driver reannealing or driver tester reannealing will get stuck, and the rest of the cDNA will pass through, and then we will clone them up. Okay. Yes. We also have discussed about array-based methods. The same thing, but array, I hope you remember, where we have a large number of spots in this case, like this. That is an array of spots here. And I think we already discussed this. Now the word there, I use the word genomics. There the sample could be the direct DNA, genomic DNA fragmented and then used on microarray. Here it is because we are trying to talk about transcriptomics. We would have taken the cDNA mRNA from two types of cells, converted into cDNA with fluorescent dyes, and then we combine and we add it to the chip. And the chip will consist of several spots, each spot representing one gene, okay, one ORF probably. And each spot, if I enlarge it, will have sequences single-stranded like this, right? And all the sequences in one spot are the same or identical nucleotide sequence. So if an mRNA is produced if in cancer cells as well as normal cells, both could bind here. That means it would have had red as well as green fluorescence. A combined color is usually visible in the microscope, and then it will give upon emissions. You are using a laser 1 and laser 2 for excitation. You will get different cells different spots excited at different wavelengths, and then a picture. The computer can overlay this picture onto this picture, because of which you will generate a final output like this, which is good for analysis. The red, a spot that is red, would imply it is produced only in the cancer cells. A spot that is green would imply that it is highly expressed only in the normal cells. A spot that is yellow would imply that it is present, it is expressed in cancer as well as normal cells. Okay, that is about transcriptomics, microarray based transcriptomics. So now we are trying to go a little further. We are interested. This is a nice experimental setup. The goal of it is to the goal is to find all the, say for example, there is a genome, and there are several promoters in this one, right? It could be in this direction or any of these. My job or my goal is to find out all the promoter sequences. How can I get all the promoter sequences? Or at least it's a kind of screening technique. So what one can do is have a probe which with the LAGZ reporter, origin of replication, selectable marker, everything are there, okay? And then you would have done all the assays in M13 or M15 E. coli that is deficient in LAGZ alpha domain, alpha fragment. So it is again based on alpha complementation. Yes. So here is the cloning site. What they have done, one of the papers, actually, I think it is from a paper. They have taken the genomic DNA and they made it into several pieces. Okay, they fragmented. And the lag set here in the vector does not have, there is no promoter here. There is a cloning site, there is no promoter. So if a cell has this vector, it does not produce lag set. So it would typically give white colonies. Now we are constructing a kind of library of all these fragments. Okay. Some fragments may have vector promoter, some fragments may not have a promoter. And if we have cloned, so there are two situations. Say this is lag set. In some cases, a promoter sequence may come here. In some cases, a promoter sequence may be like this. In some cases, there is a fragment included, but it doesn't have a promoter. So in these cases, all those colonies, if a cell is able to give rise to blue colonies, which would mean that the lag set has been expressed. Normally, as we established here, lag set is not expressed because there is no promoter in this vector. But if the same in a similar setup, if it is giving a blue colony, it means lag set is expressed. Lag set must be expressed if only there is a expression. That means a promoter sequence has been incorporated upstream of lag set. Okay. So this way, they were able to take all the blue colonies and analyze the cloned fragments and identified multiple promoter sequences from that target organism. Here, if you look, this will give blue color. This one will not give blue color because it is actually transcribing any fragment or gene on this side, not of the lag set. And those that do not incorporated ones that do not have a promoter will also give white colonies, not blue colonies. This is how screens are built. Our objective in the majority of the cases, we want to know what are all the sequences that are present, that are involved in lag set, if you say. I mean, in the lag operon. You have to build a genetic screen. And this is a good enough genetic screen for that purpose. All you have to remember is in the host should be deficient in the lag set part, right? Like we have discussed in the alpha complementation. So that is host you should remember, and in the construction of the vector, there is no promoter upstream of lag set. That is another thing that we should remember. So in today's class, this is the focus. Okay, you must have heard about it, or you may not have heard, but that's all right. Yeast 1 hybrid system, and there is another one called as yeast 2 hybrid system. You may have heard, so let's just see how far we get. Here, our objective for yeast 1 hybrid system is to figure out if a protein, we are going into from expression to we are going into function of the gene. The function of the gene product, right? A gene produces a protein and or RNA, and we want to know what does that do. Does that protein interact with other proteins, or does the protein interact with DNA or RNA, and so on? Whenever we are trying to remember or know of a function of anything, we should know with what all does that protein interact. So here is a technique called as yeast 1 hybrid system. It is to find out if a protein binds to DNA or not. Binds to a specific DNA, okay? A DNA sequence, we will put as so that it will be some specificity. I want to know if a protein binds to a specific DNA sequence. That will be the objective of 1 hybrid system. And here is the reporter. There is called as bait sequence. Bait is the DNA. So I assume that there is a protein that can bind to a particular target sequence, which has some sequence that is specific and unique. So this is called the bait. You know, right? In fishing, when you are going to fish, you will put the to the fishing line, you will put some food material, and you throw it into the pond or a river, and then fish might bite it and get hooked to it, and then you pull it up. That's the bait. So you construct a reporter like this. You have the vector construction. You have a reporter, and you use bait sequence here. Um, there is a transcriptional. I think I will only talk about. I'll see. Gal4. There is a transcriptional factor in yeast. Gal4 protein has two domains. One is called as DNA binding domain, or we will just call it as binding domain. Okay. And it also has another domain called as activation domain. So what it typically does is, we are now talking about the reporter here. So here is it is involved in galactose in yeast, and with the use of DNA binding domain, it interacts with this sequence. It's a transcriptional factor, so it does interact with the DNA, and it also activates the RNA polymerase to bind and allow the transcription of this particular gene, say Gal4. Okay, no, Gal4 is the transcriptional transcription factor. So this is the binding domain with which it interacts with the DNA, and this is the activation domain with which it activates RNA polymerase to initiate transcription. I hope that is clear, right? Now I'm going to erase it so that so now if I have a situation, a scenario like this, where this is the gene, and I separate these domains of Gal4, binding domain and activation domain, and I add it to the target sequence individually. Okay, if there is no, once I added binding domain, will there be activation of this gene? It will not be there, because it is going to bind here, but it doesn't have any activation. It cannot activate the RNA polymerase. If I added only the activation domain, it can activate RNA polymerase, but it doesn't know where to activate RNA polymerase, not specifically in this place, because there is nothing that is putting the activation domain in this location. So this kind of logic was used. But if I somehow have a protein, okay, if I have some other protein, if this is the sequence, say for example, I have X protein X, and it binds to a sequence represented like this. Okay. And if I have made a translational fusion, I hope you remember translational fusion, wherein I put X here and the activation domain of Gal4, then a protein will be produced that is having two domains, domain X and domain activation domain of Gal4. Right? This is one continuous protein. Does this protein completely have activation, has the capacity for activation? Yes. Does this protein have DNA binding? Yes. But it's not the same DNA as in the probe here, as in the promoter that was here. But it will bind to the DNA I represented with cross mark, something like this. Okay. Now we generated a new hybrid protein. We have generated one hybrid protein, and that is why it is called as one hybrid system. It is basically used mostly in yeast, or it was first reported, or I think Gal4 here used is from yeast, so we call it as yeast one hybrid system. we are making one hybrid protein, right? Hybrid protein with one protein that might be putative DNA binding and one activation domain. Okay. So now the experimental system, I hope you understood this. I'm going to erase it and try the actual get into the experiment. So now, for example, I have two proteins, protein X and protein Y. I don't know if protein X or protein Y binds to DNA. I want to figure that out. So I'm going to construct to perform two different experiments. So now this is the tester. I don't know. My goal is I want to know if X or Y binds to a DNA, a DNA that I'm representing like this. Okay. My goal again, I will state the goal. My objective of this study is to find out if X or Y can bind to the target sequence. Okay, I know a specific sequence. I want to know if it binds or not. So what I will do then is I will construct. First, I need to make the one hybrid. That is, I should have had. This is all preparation, where I will make a translational fusion between X and the activation domain of gal4, because of which I will have the generation of something like this, X and AD. The same thing I would have done in the Y, where I would have made Y and AD so that it will generate a protein that is something like this. Okay, say this is a vector and this one is the vector. They would have produced a product like this. Now I take a cell. Now I have to do prepare the reporter. I need to have a reporter. So for simplicity, let's use lagz. Okay. So the host should be lagz minus. So it will produce blue color upon using xgal. What should I put here is I'm going to put the target sequence that I said here, right? So you can have multiple copies of them. Sometimes if the fragment is not, you don't need to always, but it's not a bad idea. So what will happen now? If blue color happen, if blue color is produced upon in this cell, okay, in one cell, in one cell, now you have the vector of the for the hybrid, this one. So which has X and AD, okay, and you have another vector here wherein you are having the bait sequence and the reporter like lagz. If this cell gives white color, what does that mean? Right? That means, so from X and AD, it will produce a protein X and AD, a hybrid protein. If it can bind to this target sequence using the X, that means if X can interact with this, then activation domain will allow transcriptional factor to bind here and allow the transcription of lagz, and lagz would have given blue color beta galactosidase activity. But I'm saying it is white in color, which means that X here is unable to bind to the bait sequence. Okay. So that means, in according to my objective of the objective of this experiment is, I want to know if X or Y interact with this particular target sequence, that is the bait that is used as a bait in this. Okay. Now let's just take up Y. So I would have used the same bait. I would have used the same sequence. So in this cell, we would have the construct here, which will produce the hybrid. So it will produce Y with AD. It should produce a protein something like this, Y and AD. And it also has another construct in which you have the bait sequence and laxate. If a cell grown on in plates containing X-gal gives blue color, what does that mean? That means this hybrid protein Y and AD is able to interact. There is only one DNA binding with this, with the target, with the bait sequence. That is Y. If it binds here, and AD sequence must have activated transcription of laxate, and laxate must have given rise to blue color. You are indirectly able to figure out if the protein either X or Y is able to interact with the bait sequence. Okay, that is how one uses um, these East 1 hybrid system. Is that understood? I will stop here, and I would want to take any questions you may have. Please ask me some questions so that we will discuss. Don't worry if you didn't understand, or you are shy to ask, or you couldn't follow, because in the next class, I'm going to discuss about East 2 hybrid system. I would repeat with East 1 hybrid system. Okay. So the goal, you should understand the goal. The objective of this technique is to figure out if a particular protein binds to a particular sequence or not. Okay. Any questions? Hello. No questions? Okay, then what I will do is I have some things to say with regard to assignment.